Isabel was feeling a little nervous, so I will go and stand there first before she comes out. We are here for one of the institute level seminars. This will be given by Dr. Isabel Vales, who is the principal pigeonpea breeder. Isabel has been with us since July 2010, so we are almost 2 years, in another 4 months to go. And considering that she has spent a year and a half with us, looking at all the activities in pigeonpea breeding, and also looking at the fact that we are also considering this as a succession planning for the father of pigeonpea, K.B. Saxena. We thought it pertinent to request her to give a seminar in which she can indicate what she has learned, the lessons learned in the pigeonpea breeding in the last 1.5 years, and her perspective of where the pigeonpea breeding at ICRISAT will be in the next 5 to 10 years. So Isabel will be giving that seminar when I finish this. Isabel did her initial degree, undergraduate degree in biological sciences at the University of Santiago de Compostela in Spain. Her graduate and doctorate degrees in biology also in Spain, but at the University of Vigo. And she worked as a postdoctoral research fellow at the University of Minnesota in the US. And during the time when she was working in the university in the US, her work was mostly on both conventional and molecular approaches to improve the genetic studies in maize, wheat, and barley. And in August 2005, she was hired as the head of the potato breeding and genetics program at the Department of Crop and Soils at the Oregon State University in the US. And she has been a team member in the release of 15 potato varieties during the time she was there in that position. With this brief introduction, I would now like to request Isabel to give the seminar. As indicated to you, Isabel, you can take about 40 minutes for the seminar, and then we will have 15 to 20 minutes for discussion. Please.
Thank you very much for the introduction. Thanks to everybody for coming this afternoon. And without further ado, I'll start right away because I think I have, prepared quite a number of slides and see how far I can go. Otherwise, I'll cut some of the pieces at the end. The title of the seminar is Pigeonpea Breeding, Challenges and Opportunities. And I'll cover aspects related with pigeonpea production, setting priorities also, and breeding goals. What are the main constraints in pigeonpea production? Also, how to combine conventional and molecular breeding aspects, ensure the success of the pigeonpea hybrids, aspects of data management, and then just a few words about funding, and finally what kind of outputs would come out of the program. In terms of pigeonpea production, the graph here represents the worldwide production, and like you can see, area per se and production have been increasing over time. However, yield has been quite stagnant. The total world area in 2010 was 4.8 million hectares, and in India it was 3.5 million hectares. India produces 70 to 80% of the Pigeonpea. However, it probably consumes near 90% or more. So there is not enough Pigeonpea produced in India, and we import Pigeonpea from places like Myanmar and other areas in Africa. The first thing when you think about the breeding program is setting up the priorities in terms of what would be the target area, target environments, what kind of germplasm base, selection criteria you use, type of cultivars, and also what are the roles and responsibilities of the institute per se, and as well as all the partners, because this is not just ICRISAT alone. In order to set up all of this, we need to take into account the situation analysis and also the desired outcomes that should match quite well with the institutional outcomes. We are now in the process of preparing the Pigeonpea strategy for India. I'm leading that effort, and then the same thing is happening for Africa, so we are defining this in more depth. In terms of breeding goals, the main thing is to increase yield. Increase yield that has to be stable over locations and years, and also maintaining the grain quality. At the end, developing new varieties with higher yield that can be used later on either as varieties or as parental lines of the hybrids. And some of the aspects in order to prioritize the specific objectives or the selection criteria are related with the agro-ecological zones that we are targeting, also the maturity groups and phenology of the crop, and also the final use. In terms of agro-ecological zones in India, for Pigeonpea, there are four major agro-ecological zones that are listed here in terms of importance. And those agro-ecological zones are defined by edapological considerations and climatic considerations. And we also should consider aspects of how is the crop planted? Is it intercrop, sole crop? What are the cropping windows? And also what are the main constraints in each of those agro-ecological zones in terms of pests, diseases, and abiotic stresses. In terms of maturity, Pigeonpea is a really amazing crop because the range is very wide. If we go by flowering time, we have now varieties that flower in less than 50 days and others that need more than 160 days. And the preference for maturity is also region specific. Some places they prefer short duration, others medium, others long. Flowering pattern is the main two types that are described, even though we talk about some intermediate aspects. The main are the determinate one, where the growth would stop at the flowering time, and then you have the indeterminate one when it continues growing. In most cases, the indeterminate would be preferred because it receives less damage by the insects, but we are still putting effort, even smaller, indeterminate types that are desired in some areas that would be more suitable for mechanical harvest and synchronization of maturity. In terms of the use-specific traits, depending on what is the final use of Pigeonpea, and Pigeonpea is really an amazing crop for sustainable agriculture because it has lots of different uses. But considering just a few of them and taking into account the final use would determine what traits we need to target. For example, if the final product would be processed dye, we need to pay attention to the milling quality and the seed size and seed color. If it is for fresh or canned vegetable, also, we consider seed size, seed color, pod length. In the case of using the Pigeonpea for feed, then protein becomes like an important trait. And if it is for fodder, then returnability and biomass would be traits that we need to consider. The basic breeding approach, and this is kind of simplifying things quite a bit, but so it's straightforward. Identify lines that contain the traits of interest. This would include even the cultivated lines, varieties, that people really consider the most adapted, the better ones. Also, advanced breeding lines, land races, wild relatives, to make crosses to combine the traits of interest, generate segregating populations using the appropriate breeding methods, and then we apply selection. We can use conventional and marker-assisted selection procedures to reach our target. And also it's very important to do agronomic, quality, disease, pest-resistant trials across locations and across years. In Pigeonpea, we are quite lucky. We have many genetic resources, and even right here with us, there are more than 13,000 cultivated accessions and more than 500 crop wild relatives. As well as for the cytoplasmic nuclear male sterility system, right now there are 7 different cytoplasmic bases for this system. And we can also use mutants that will include even dwarf types, different shapes of the leaves, of the flowers, etc. Within ICRISAT, the germplasm is being sorted or accordingly, so we can have different levels of depth. And so they have divided into like the active collection, core collection, mini core collection, and base collections. And these collections are being studied in relation to traits, so we have better understanding of what kind of interesting genes could be there. Some examples of the wild relatives that carry useful traits are listed here. There are many more, and in some cases these wild relatives have been crossed with the cultivated ones to develop what is called the crop wild relatives introgression lines, which would be very useful for the breeding program. In genomic resources also in the last, let's say, 5 years, lots of things have happened, and we should be very happy that now we have more tools at our disposal, including more than 25 mapping populations, also killing population. Molecular markers, quite a number of them, more than 3,000 SSRs, more than 700 SNPs, and more than 1,500 DACTs. Also, there are back libraries, and even the whole genome of Pigeonpea variety Asia has been sequenced, so that is available right now. And also, there are good transformation capacity available. In this table, we have listed just a representative set of some of the mapping populations that we are maintaining within our Pigeonpea program, and some of them are quite advanced. We just harvested a seed out of the top 3. And the top 2 or 3 will be using, during this year, we'll do a phenotyping for, the second one and the third one. We'll do phenotyping for Fusarium wilt and Sterility Mosaic disease, and also together with the genotypic information for marker trait association studies. Now, in terms of increasing the genetic diversity at the nuclear level, this is really a big need, because we claim that the base we are working with is quite narrow. So, like I have indicated, there are a number of things. So we have crop wild relative introgression lines, land races, mutants, elite lines, parental lines, varieties. Lots of things are there. But the breeding program cannot just take all of them directly. Some way of filtering them. So we only use the ones that have the better chances of being useful. We need a way of filtering. One possible way of filtering is with all of them, you do association mapping, for example, that would be really useful to study marker trait association, but also to identify those lines that have more potential for the breeding program. And in terms of the mating and selection schemes that we could use to increase the genetic base of the breeding pool, as well as to enhance recombination, and also for genetic studies, include a number of aspects like biparental crosses that could go directly selfing without selection all the way to recombinant inbred lines, or with selection at each of those states. So biparental crosses are pedigree based, or we could get one back cross followed by selfing, and then later on proceed with selection of multiple rounds of back crossing and to introduce the traits that we are interested in. Also, to increase the genetic base of the breeding pool and enhance recombination would allow us to identify good combinations of use for the breeding program, and also to do genetic studies. One of this is multi-parent crossing, and this is called the magic population, multi-parent advanced generation intercross, that would involve starting with 8 quite diverse parents and do 2-way cross, 4-way cross, 8-way cross, and then move by selfing all the way to recombinant inbred lines. Another multi-parent type populations, what we call non-population, nested association mapping. This is again another place where both interest of the molecular genetics and interest of the breeders come together. Because this type of population is useful to understand the genetic basis of the traits, but it's very useful also to generate lines that contain new combinations that might be of interest to the breeding programs. In this case, Asha, which is the most popular medium duration variety, would be a common parent, and it would be crossed with a number of diverse lines, and after that generate recombinant inbred lines. The diversification at the cytoplasmic level, we have indicated there are already 7 different types of CMS, this is the list of wild species that are involved in this type of CMS. Even we have the A5, the female line is in Cajanus cajan cytoplasm. Out of all of those, we are studying them, but up to now, the one that has shown us the most stable one and the most promising one is the A4 CMS. Because the A line has male sterility that is stable across locations and across years. There are also a high number of fertility restorers to make the F1s, and also the fertility restoration of the hybrid is pretty high. One of the aspects in terms of moving along with the breeding program is to understand, out of the traits that are important, what is the genetic basis of the traits, and use that into consideration at the time of developing the breeding schemes and breeding programs. So we need to know how, is this a qualitative or a quantitative trait? How many genes are involved? What is the location of the genes and the position? What is the gene action? What are the interactions? Are all those intrallelic, interallelic, genotype by environment interaction? Also, what is the heritability of the trait? And what is the percentage of the phenotypic variant that we are explaining with the markers that we identified associated with the traits? In order to estimate the number of effective factors, there are classic methods that can be used, and then there are also like QTL-based approaches that we can use for marker trait association. I'm going to describe some of the important traits for us in the program and a little background of what we know and how can we use some of this for the breeding program. One of the things is flowering pattern. Flowering pattern is quite important. I have indicated that the NDT types seem to be better in most cases because they recover from the damage caused by the pollen borers. So NDT types, non-determinant types, are preferred over DT types in most areas. One major gene has been identified as differentiating DT versus NDT, but there are also some minor genes that modify the expression. And NDT is dominant over DT. So in this particular case, pedigree and backcross methods can be used to obtain the lines that we want, either NDT or DT. And one possible use is that, because we know that NDT is dominant, if we have for the parents of the hybrid one parent DT, one parent NDT, that's one way we can monitor that the parental lines are pure lines. The DT line, all of them should be DT. We should not see any NDT. So it can be used as a morphological marker for testing for homozygosity of the parent. Now, the knowledge of the molecular basis of these traits would also be quite useful, because that way we could identify heterozygotes and confirm homozygotes at early generation stages, and that would facilitate our selection process. Flowering time and photoperiod. The flowering time and maturity are region specific. And for example, in the central zone, south zone is mainly prefer medium maturity, while on the western zone they prefer short maturity, eastern zone it could be short or long duration. So it's region specific. We know that flowering time is highly heritable, and we know that earliness is partially dominant. We also know that in general, earlier flowering genotypes tend to be less photoperiod sensitive. And the photoperiod aspect is quite important, because I really believe that photoperiod sensitivity is one of the things that has prevented Pigeonpea from expanding all over the world. And for this photoperiod response, based on knowledge from other legumes, it's likely that two major genes are involved, so we should be able to target them. Because of the nature of this trait, pedigree-based selection can be used and has been used already. We have to report that lines that flower in between 45 to 50 days have already been obtained and are very advanced generations and performing very well in places like Punjab and in Almora. They are very happy with those. The knowledge of the genetic basis would allow us to select at earlier stages, and also would allow us to introgress photoperiod insensitivity in medium duration plants. And the maximum yield capacity is within the medium duration, so introducing photoperiod insensitivity there would make a lot of sense. Fusarium wilt, we know that major and minor genes for resistance or tolerance have been identified. Also that resistance is dominant over susceptibility, but also susceptible parents can carry minor tolerant genes. Now the major genes can be introgressed via pedigree and by cross approaches. And because of this fact that the resistance is dominant, if we want to generate hybrids, with having only one of the parental lines would be enough to develop resistant hybrids. So we could have the Fusarium wilt in the A or B lines, for example, and that would guarantee that you have a resistant hybrid. It would be, in any case, very important to know marker trait associations to better target the genes involved, also know the position, effects, and interactions. Because we also know that up to 5 races have been reported for this Fusarium wilt, and depending on the races and depending on the genotype, we get different messages. So in order to target things properly, molecular tools will be really important. And also for pyramiding the major and the minor genes, and that way ensuring durability and stability of the resistance. Sterility mosaic disease, also major and minor genes for resistance have been reported. In this particular case, resistance to SND has been reported as recessive in some cases and dominant in other cases over susceptibility. And resistance is isolate specific. Major genes can be introgressed via pedigree, base and backcross approaches, but selfing will help to expose the recessive genes, the recessive alleles. When we say in many cases resistance have been reported as recessive, we need to self-expose the genes. And it would be better if all the parents of the hybrid are resistant. Also, marker trait association would be quite useful, and in this particular case, SND already there are at least 2 reports of marker trait association that we can follow. But we know that there are at least 3 different isolates for this virus, and there is a different specificity. So we need to take also that into account, that it can be isolated, the resistance can be isolated specific. So in the context of climate change and all of these things, if some of the more aggressive isolates start coming towards us, we need to get ready for that. For example, the Bangalore isolate seems to be quite aggressive. So we need to be sure that we build up the resistance for the, let's say, Hyderabad, Bangalore isolates together, and put them together so also to ensure the durability and stability of the resistance. Pod borers, everybody knows pod borers and pests are the main problem in Pigeonpea as well as in many other legumes. Host plants resistance is actually scarce. There are not many lines that have shown tolerance. There are a number of wild relatives that have been reported as having tolerance or resistance to the pod borers, both Helicoverpa and Maruca, and also crop wild relative introgression lines have been developed, and we are trying to incorporate those in the context of the breeding program. The mechanisms of tolerance or resistance have been determined as being of its positional non-preference and recovery capacity. We also know, as I indicated before, that the entity types suffer less damage. So we are trying to focus on that, on the entity types. Transgenic BT is probably one of the best options to help with this problem. Already there are reports of confirmed effective transgenic resistance. So thinking from a breeding perspective, one of the approaches that would make more sense for us is already use those events that are successful events and backcross those using marker-assisted selection to the parental lines. So that will introgress the BT gene into our parental lines for then developing for border-resistant hybrids. The other thing is also we can introgress the BT gene into varieties. And in the case we worry about gene movement, we have a type of flower that is called Christogamous flower that doesn't open, so in most cases is fully self. That would be an ideal thing for putting this BT gene into this type of variety, so gene flow might not be that much of an issue. And at the end, what I really think is BT is likely the best option, but we need to go on the safe side. So combining some of the partial host plant tolerance with the BT would really ensure that there is long-term resistance for the pests. In terms of yield and yield components, we know that both additive and non-additive effects are involved in the yield and yield components, and that the yield components that show the higher levels of heterosis are pods per plant, pod clusters per plant, branches per plant, seed size, seeds per pod, and plant height. So we should proceed with the improvement of the parental lines for yield and yield components, and this mainly take care of the additive effects, and then development of the CMS hybrids that would benefit from both additive and non-additive. And at the time of developing the CMS hybrids, focus on the performance per se, heterosis, and also on the specific combining ability. Within the abiotic stresses, one that we are working on right now is waterlogging. I think drought might need to be added because terminal drought is also very important in Pigeonpea. We know that resistance or tolerance to waterlogging is dominant over susceptibility, but likely based on the evaluation of a large array of genotypes, we believe that also minor genes are there because there is a continuous distribution. The major genes can be introgressed via pedigree-based approaches and back-process approaches, and because of the fact of being dominant, if we have the waterlogging tolerance in just one of the parental lines, let's say the A, for example, that would really allow you to develop resistant hybrids. In any case, also, we are quite interested in studying the marker trait associations to go more in depth into the genetic basis of the trait, and if we have a number of minor genes in addition to these minor genes also combined, because again, durability is the key, and stability. In this graph, I'm trying to put together a number of things, and in terms of sources of sources that we can use for the breeding program that include breeding lines, cultivars, land races, crop wild introgressions, mutants, etc. That can be, at times, some of these can even directly be used and select out of the diversity that already exists there for moving into the system of developing new lines, or otherwise used in biparental populations, either pedigree-based populations, backcross populations, or in multiparental populations like the ones MAGIC or NAN, to generate large populations and then phenotype them, genotype them as well, if we can, and then proceed with selections. We would generate a number of lines. Now, in order to figure out what is the best approach for hybrid development, identifying parental lines and hybrid development, lines selected, we should involve them in test process and in diallele process, and take a look at the specific combining ability. Based on the specific combining ability, we can separate lines that combine very well, that could be going into two different groups. The group that would be for the B lines and the group for the R lines, if they have high specific combining ability. Now, in the case of the B lines, of course, we always need to get the A lines, so that we need to do the backcrossing approach to generate A lines. And then by combining the A line and the R line, we'll get new CMS hybrids that ideally will have the higher level of heterosis, because we took into account specific combining ability to segregate the lines that should go into the restorer group versus the AB type group. I really think in Pigeonpea it's time to move into molecular breeding, and there are a number of advantages. One of them is early selection. The other thing is selection in absence of the disease or in absence of the trait that you need to evaluate, or without doing any test even for fertility restoration. And it's also independent from the environmental conditions, and it allows us to pyramid multiple resistant genes. All of that would reduce time, would reduce the number of lines that we keep in the system, and would also guide selection. Of course, there are always challenges. Some of the challenges involve aspects related with lack of recombination, having traits that are very complex that we cannot really target with the markers, and also maybe even some crossing barriers. But definitely, I think many crops have followed this path. I think Pigeonpea is getting ready to follow that path as well. In any case, phenotyping is really essential and continues to be the most important aspect. No matter how much we do at the molecular level, if the phenotyping is not accurate, we will not get a good message. So we need to combine phenotyping and genotyping very carefully. Now, I have indicated about developing lines that could be used either as varieties or as parental lines or hybrids. I have said that because in some cases, hybrids might work very well, but maybe in some other places it would not work as well. And some of the aspects for consideration are involving like seed production. Not all the places are suitable for production of the hybrids. For the hybrids, we really know that pollinators are required. If that is not there, then you cannot produce seed of the hybrids. Also, can you access fields that are in isolation, more than 1,200 meters from another field, yes or no? If not, then you cannot produce hybrid seed. Also, technical expertise for hybrids is higher, the cost is medium to high, and the importance of the pure genetic stocks is really essential. And the seed system needs to be formal or well-trained informal. And in any case, for commercial production, in order for the hybrids to be of benefit to the farmers over varieties, the heterosis and for yield has to be at least more than 20%. In any case, for those situations where things can be arranged, hybrids are the great promise. High yielding disease tolerant hybrids, that's one of the biggest achievements in the Pigeonpea program, because there are lots of advantages for the hybrids. In terms of yield, 30 to 40% more yield, more bigger. Also greater shoot biomass and root biomass, which would be very good for reducing the seed rate and also for drought tolerance. And the hybrids are ideal for intercropping and also give the farmers more wood, which is another use of Pigeonpea that the farmers really appreciate. I'm going to show you just the top 3 hybrids within the program. This first one, ICPH 2671, has been officially released, and over a period of 4 years, a total of 43 multi-location trials, the yield at advantage is near 36% over the medium duration variety Maruti. So that is a very good one, has been already released, and many people are quite happy with it. The other promising one is ICPH 2740, for which variety release is in progress. And over a period of 4 years, a total of 33 multi-location trials, the advantage in yield is close to 38% over Asia. And this one has the best chances likely to move forward because the seed color, everything is very, very well accepted by everybody. And a short duration one, ICPH 2433, also over a period of 3 years, 33 trials. The advantage over the local Czech for short duration UPAS 120 is near 32%. And here you can see a picture of this hybrid ICPH 2433 out of our station here in ICRISAT. Just checking the amount of flowers, you can probably predict the yield. It's really very good hybrid. Now, in terms of ensuring the success of the CMS hybrid system, there are many things that need to be considered. One of the most important ones is the purity of the parental lines, but also we need a stable male sterility system. Be sure that the heterosis is very good. Also, professional implementation of the CMS hybrid technology, constant training, monitoring, and evaluation. Also, efficient formal and informal seed systems, and marketing and promotion efforts. There is a big challenge for the hybrids. By 2014, KB needs to be sure that the hybrids are in 100,000 hectares. So, big challenge there. Now, I said that parental lines and pure parental lines are really the key for the success of the hybrids. What could cause problems or impurities with the parental lines? Outcrossing is one of the main ones. Mixtures, also a possibility. In some cases, non-stability of male sterility system, damage by pests or diseases, also even de novo variation, unintended selection, or even early release of heterogeneous or heterozygous parental lines could cause that. But again, this is really critical, and we have even went through the effort of developing a checklist that has many different points that need to be ticked to be sure that you reach success with this. And I'm going to only talk about those highlighted ones. Genetic purity, talk about seed production in terms of isolation and the presence of pollinators. Genetic purity. The typical way of testing for genetic purity is about visual observation under field conditions. So you take a look at the aspect of the plants and the morphology, uniformity of height, health, and also at considerations of fertility versus sterility, and draw out the of types. The other thing which is novel that we are implementing, and this would be very useful for the farmers, is what is called naked eye polymorphism. There is one mutation of the leaf that is called of corded leaf, that is when you have recessive alleles, you see the off-corded shape. We have transferred this trait to A and B lines. So now we have A and B lines, all of them should have this off-corded trait. Otherwise, they are not pure lines. And when you cross with the normal restorer, the hybrid would be having normal leaves. So this is very good. There are some other leaf morphology that we are exploring. Because the morphology you can tell right away. The other aspect is using molecular markers. Check polymorphism between the A line and B line, and then use the markers that would be diagnostic to confirm hybridity. That is also a very good option that we are already implementing. For seed production, isolation is really critical. Depending on what stage and how much seed you need, you could go just by simple bagging of individual plants, or using nets that don't allow the insect pollinators to go in there, or isolation fields with more than 500 meters from another field. Pollinators are really essential, especially for hybrid seed production, and it could be, you could pollinate under field conditions naturally, or are there artificial conditions, and they're using cages where you can have hives, or even, sometimes we use our technicians as pollinators. Important considerations in the fields especially are that you have a good insect population and there is water, trees around, and the weather conditions are also pretty good for the pollinators to feel happy and do the job. We have done a couple of years of experiments of pollination using net cages. There are some good things that we are learning out of this, but also I think the system is not yet perfect. We have Apis mellifera, the Italian honeybee, quite sophisticated. Initially it doesn't like the pigeonpea, takes some time to get adjusted. We now know that at least they visit the B lines 2 times more often than the A line. Some adjustment still is needed to be sure that they do the right job, or maybe we need to check for some other pollinators. But that is another option if isolation is not acceptable. No matter what, for seed production of the hybrids, some locations have been identified as good locations, some bad locations, and in many cases the presence of the pollinators is one of the key factors. So those are things that we can recommend if somebody is interested in producing seed. We can indicate, okay, don't do it here, do it in these other sites. In any case, for the success of the pigeonpea hybrid production, the purity of the parental acid is essential, and now we have tools. Those SSR markers can be used very good for testing for purity, as well as the incorporation of phenotypic markers like this naked eye polymorphism in terms of leaf shape. Could I continue a little more? 5 minutes? Because this is cut a little bit. There is one new area that I really wanted to push hard and is related with data management that I think is really essential. And so that requires trained personnel, modern equipment, and also a data management system. This would really allow us to produce, get data and results faster, and also facilitate the decisions in breeding. Trained personnel is really essential to move on this system. Younger people are great. They already know a lot about computers, very comfortable. We really should hire more of them. Modern equipment, we have purchased some of these things. We are in the process of purchasing some others, like single plant threshers, dial processors, grain counters, data loggers, barcode readers, balances, etc. And this actually became like a very important aspect when we had problems with TFL. We said we need to go more into mechanization, more automatic data collection, because that would really be of great benefit to us. Now, in terms of this data management software or data management system, there were several things that were required. One was that it was a relational database that we could have phenotypic, genotypic data. It could allow us from the moment of designing the experiment, crossing blogs, field books, data collection, sharing data, analysis, even for searching, decision tools. It should be friendly and have different levels of control. Some of you have been involved in the process of searching for data management systems, and after evaluating a few, we had decided to purchase AgroBase, so that's the one we are using right now. It took a while, and we're still working on standardization and everything has to be standard and uniform for this system to work properly. It requires time. When it works, then it's quite simplified. So we have to work quite a bit on a number of aspects, including trade dictionaries, etc. Now, having a system like that is really wonderful because you can have inventories of the crossing books, inbred lines, segregating populations, everything that you want to store there, multi-location, multi-data, easy accessible by all of your colleagues. Immediate benefits. You can save time, money, improve efficiency, and reduce errors. For example, for data capture, just collect directly and then transfer directly to the computer. You don't need assistance for going from paper to computer. Some curation and cleaning of the data is really required, but there are also some validation tools that we can use. Templates, once they are ready, you can reuse them. You can save paper and ink. And additional benefits is that the data is immediately available for analysis. The data analysis is simplified because it's connected with data analysis software like GenStat, and also it allows you to make faster decisions. Okay, we'll cross this one with that one because we already know it's good. Some of the outputs of this data management includes new discoveries that can be based on exploring old data or historical data. information for use in new data-driven research, preserving the scholarly outputs, facilitate data sharing, but also facilitate queries. If I want to know how many lines have resistance to SMD, Fusarium wilt, and Phytophthora, I could just do a query and come up with that. And also more and more, this is the requirement of some of the funding agencies, because they need data management, also retention of the data and access to the data. This would be what we are doing with AgroBase is linked or related to efforts by the Generation Challenge program in terms of this integrated breeding workbench that goes from the moment of planning the experiment all the way to the final decision making. And now with more of the molecular tools that will be also integrated into the system, so all of the pieces will be there. Phenotypic information, genotypic information, data analysis to really make faster decisions on this. Okay, in any case, I'm going to kind of wrap up with some of the outputs that could come out of the Pigeonpea program. And this is not only ICRISAT, of course we have the partners at the national system, international NGOs, etc. But some of the things that are coming out of the program and should continue coming out of the program include improved pure lines that can be released directly as varieties, improved parental lines. Also for the case of the hybrids, because we want to know more about the heterotic groups and about fertility restoration, we should be able to identify the best hybrid combinations much faster and efficiently. Also genetic knowledge of fertility restoration, heterosis, Fusarium wilt, SMD, and waterlogging. The CMS hybrid production technology, it's already well established but needs to be shared with the partners. Molecular breeding needs to be implemented in the system. We have initiated some aspects already. Data management, I think, is essential and we'll continue with that. We're also well placed for doing formal courses in breeding, genetics, and seed production, as well as for training technicians, farmers, graduate students, and scientists. And one of the aspects that is coming more and more now is the consultancy demand. In terms of funding, I'm not going to say too much, but just a couple of things. Is that being Pigeonpea a crop that is not worldwide distributed, I have found myself sometimes difficulties of getting international funds because they say Pigeonpea is not a crop of importance to, a country. The way of going around that to be able to get international sources of funds is to focus on climate change, things like drought, waterlogging, and also in terms of poverty alleviation, whatever is linked to technology transfer. In that case, I think international funding is possible. And we have lots of support from the Indian government. For that, we can continue on that regard in terms of we can push on that the molecular genetics, molecular breeding, and also the expansion of the hybrids. And thank you very much. I think with that. Just in case if you are wondering about this picture, this is a new use of Pigeonpea for art. The head is made out of Pigeonpea, the yellow part.
Thank you, Isabel. Now we will open up for questions, any comments, and please make the questions targeted and short, and so many people can have opportunity to ask questions. Can I get an indication who wants to start?
Start with prosthetics. I think two of the things you mentioned we have been discussing in the past. One was the integration of DT, of course, into the AT line. So we need to make some sort of a decision on how exactly to pursue that. And it's not the optimization work we will discuss most of the. And second, I think I'm more curious about, we discussed when we joined earlier, was using some sort of a marker system like histogamus flowers or something else, maybe flower shape for inducing for pursue this further. But I'd like to hear more what are the opportunities available, especially using histogamy or morphological trait to associate with transgenics?
I definitely think that having some type of morphological marker could be very useful to confirm that you have the transgenic. It would help certification agencies as well. Both, but that is only for varieties. It could act as a marker to identify that you really have the transgene, but it could also act as a way of preventing gene flow, because it would be almost close to 99%, 100% selfie. So for varieties, if varieties is one way, Clistogamous is definitely the way I think to go. And the leaf shape of this of corded, for example, we have already A lines and B lines. We have not committed them yet to hybrids. So if we are sure that BT Pigeonpea is there, we can reserve that type of trait for that specific use and try not to use it in other contexts.
That's a decision which we made in some level.
What is the scope to improve the harvest index of Pigeonpea, because it's very low and probably one of the target to.
I think you are totally right. You are on the dot there. Increasing harvest index would be really what you really need for increasing yield as well. So we should definitely take a look at that.
Isabel, you made a mention that B lines will be identified with high specific combining unity. Is that right?
I have indicated that based on the diallele crosses and on the line by tester crosses, you check the specific combining ability. Based on the specific combining ability, you could divide the lines involving the hybrid into two groups based on the specific combining ability. So it could be into the R group versus the A/B group.
Because the specific combining unity by definition is for the combination.
Yes.
And not for the lines. Not for a particular line, because if you see, by definition, it's the deviation from expected combining ability for a combination. So in that case, because the specific combining ability would be for hybrid combination, not for a line.
Yeah.
For lines, it's general combining ability.
Right. No, it's for lines, but I'm talking in the context of the hybrid. Once you make the hybrid, you know which two lines that are involved in the hybrid that give you the highest heterosis. So then you can, based on that, the division of the lines, start thinking about heterotic groups.
And give opportunity for Tarta and then second time.
You have shown that, that hybrids are 30% higher yield. I thought it's also important to look at its acceptance along the value chain, in the sense of traders, wholesalers, processors, and ultimately the consumer. Price is one of the indicators which will tell you whether it is acceptable along the value chain. So in that context, I just wanted to mention that the trades that you mentioned, you have not really mentioned market preferred trades, which I thought was missing. For example, Pigeonpea, cooking time and taste, and some of these, the pericarp on top, the hull, how thick it is. Some of these things are traits which the market looks for. I think in the breeding program these should be built in.
Definitely, consumer-driven traits are really essential. This is one of the things that also came in my previous job, that initially we were only based on or using traits that are of preference for the farmers. But later on, I think these things come once the projects mature a little more. You start focusing also on what are the consumer preferred traits. At this point, because of the cost involved on those evaluations, typically we only do those evaluations with materials that are quite advanced. But likely we should start earlier on, so we would not keep in the middle things that are not that acceptable. Up to now, I don't think we have encountered situations where some new lines or hybrids are rejected because of the fact that they don't have those traits preferred by the consumers. But definitely we should take a look at that. Actually, I have to just say, today I went to see how our new dye machine is working. So we'll be looking into, dye recovery and all of those things. We have also distributed bags of dye and with questionnaires for getting feedback. But if we see the need and there is consensus that it is very important, then we have to more systematically be sure that we first listen what the consumers need to say in terms of traits that should be considered, and then actually perform those tests to have the information.
Sure.
Isabel, you mentioned that photoperiod sensitivity influences the spread of pigeonpea. How it influences, that is one question. Number two is what specific measures you or your group is taking in addressing the seed production issues in the current CMS hybrids.
What?
Seed production issues. Hybrid seed production
issues. In terms of photoperiod sensitivity, the thing is that if you take medium duration or long duration hybrids to higher latitude, let's say even to Europe, 42 degrees north or whatever, they would not flower or mature, or it would take forever to flower or mature. So obviously they would not fit the cropping systems there. On the other hand, if you have materials that are less photoperiod sensitive, they could be planted at higher latitudes and could be expanding. That is just my guess, because in Europe or in the US, you don't talk about Pigeonpea, and I was wondering why. And then the thing comes, okay, even though you bring it, it would never flower, it would never mature. So that is just one of the possibilities there. In terms of seed production issues, what measures we are taking to ensure that that is properly done? We have a seed production expert that could probably further elaborate on that. But part of that is the training, get involved with the farmers and with the private sector so they really know how the CMS system works and what are the precautions that need to be taken. And also we interact with the farmers and with the seed sector in terms of monitoring, evaluating, and being sure that that is happening properly. For the varieties, what has been working quite well is the one village, one variety concept. We are wondering maybe something like that, similar, could be implemented for the production of hybrids. Any other thing, Myers, you want to add?
Okay, we should not trouble Meyer with
He's KB.
Thank you. I think we learned the last 4 or 5 years. So we have been troubling ourselves a lot. Now we know where to grow. See, for example, we have identified many locations in Madhya Pradesh where pigeonpea is not grown at all. It's not a crop there. But once we grow hybrid production, yields are more than 1 ton per hectare. Unbelievable yield we are getting repeatedly there. So that's one thing we are doing. Second thing we are doing, we are putting more emphasis on purification of breeder seed here. And we want to track initially. For example, next 5 years, the challenge Isabel is very high with us to grow pigeonpea in 100,000 hectares and with quality. So we have to do really a good job on that. And we have trained a lot of people in our collaborator universities and all to produce high quality parental seed first and then follow these other qualities and go for large scale testing. But training is very, very important. Next week also we are getting some trained farmers here for training purpose. So we are doing all these stages, our training activity, seed activity, then also we are trying to use the molecular marker now. So we have already talked to Rajiv, and then he's working out cost, how much cost it will involve. Basically we'll do primary seed characteristics through that, and then definitely other things will be followed as in other crops. Thank you.
Dr. Hari Sharma, and then Dr. Neelam. After that, the DG will have the last word.
Okay, Isabel, I'll resist asking a question about pot mowers, but you
Last word from
Very
excellent presentation. He has scanned the environment, the science in Pigeonpea breeding, and looking at the future, some opportunities. And it's a good review of the literature, okay? But I also would like to start from where Legumes ended, because I was going to expect, how do we plan, what are the next steps in terms of very tangible R4D for Pigeonpea. So it is not a challenge only to Isabel, but the whole team of Pigeonpea. Okay, so I would like also to mention this early, that my message today will not only refer to Pigeonpea, but the rest of the crops that we are working on, because I would like to prove you, because today we have a new strategic plan. And the framework of this strategic plan is inclusive market-oriented development. I hope we have put that into our hearts and minds already. This is the second year of implementation of this plan.